Okay, now we're going to continue to inhibitor, which is actually the last subtopic under the biocatalysis, the inhibitor. Okay, so before we start, actually, what is inhibitor? If you still remember, in the six factors that cause the six factor that actually affecting the enzymatic reaction, the last two, one of them is the presence of cofactor will increase or enhance the enzyme catalytic reaction. But when there is a absence of inhibitor, they also will increase the enzymatic reaction. And then vice versa, if you go when inhibitor is present, the rate of the enzyme reaction will reduce. So inhibitor is somehow to be the molecule that able to reduce the rate of enzymatic reaction. Okay, so how they will reduce it is by changing the by affecting the active site. Okay, so we know that all the enzyme they want to carry out the enzymatic reaction, they must have what? They must be able to bind with the they must be able to bind with the substrate, right? At where? At the active site. Okay, so at the active site. So meaning that you must have your enzyme bind with the substrate at the active site. Okay, so when the inhibitor is present, okay, let's say we change the color. When the inhibitor is present, some of the inhibitor, when they bind to the enzyme, it will eventually trigger the changing of the active site. So once the active site conformation is changed, does it still available to bind with the substrate? The answer is no, meaning that enzyme cannot run their function anymore. So this kind of molecule, we call them as inhibitor. And this situation that reducing the enzymatic reaction is called inhibition. Okay, it's called inhibition. So what is the difference between inhibitor and inhibition? They basically refer to the same situation, but one is the molecule that actually cause the process inhibition take place. Inhibition is the process which involve the reducing of the rate of enzymatic reaction due to the presence of inhibitor. Okay, in order to bind with the enzyme, so inhibitor can bind to the enzyme at two different locations. One of them is bind directly to the active site, okay, and another one is actually bind to any other location at the enzyme. So we call this location as allosteric site. Okay, so this is the two side of the enzyme. So the location that able to bind with other molecule other than active site, we call them as allosteric site. Okay, basically there is two type of inhibitor that can temporarily bind with enzyme. Okay, so remember inhibitor we have the same as cofactor, they are temporary bind to enzyme, or they can also permanently bind to enzyme. Okay, permanently bind to enzyme meaning that the effect is permanent lah. Okay, if they are temporarily bind to the enzyme, then their effect is temporary only. So in the temporary binding inhibitor, we have two types: competitive inhibitor and non-competitive inhibitor, based on different site that they're binding. If inhibitor bind to active site, they are called competitive inhibitor. If the inhibitor bind to the allosteric site, they are called non-competitive inhibitor. So now we look at why. Okay, so why we say it like this. Okay, so why we say that they are competitive inhibitor. Okay, so if you have the active site, example, if this is the enzyme, if you have the active site, okay, so if this is a substrate, so substrate and active site, they were able to fit together. Okay, so this is substrate. But if today I have an inhibitor which is similar shape as the substrate, also can actually enter this particular active site. Now, molecule inhibitor and molecule substrate will going to have the competition. They will actually undergo a competition because both able to enter this particular active site. They will have the competition to see which one is highly or likely to bind with the active site, correct? So if let's say today I have a lot of inhibitor and then very small amount of substrate, then definitely the competition will win by the inhibitor, so the rate of the reaction will reduce. Okay, so that's why this particular competition is occur, so that's why this inhibitor is called as competitive inhibitor. Okay, so kenapa competitive? Kenapa bersaing? Bersaing sebab mereka dua bertanding antara satu sama lain untuk masuk ke dalam active site, maka competitive inhibitor. Satu lagi adalah inhibitor yang tak perlu masuk ke active site. So if this is a substrate, so what does the inhibitor do is the inhibitor bind to the allosteric site. So when they bind to the allosteric site, they simply just change the shape of the active site. They change the conformation of the active site. Example, they become round in shape. Okay, so now when they bind, active site change conformation. So when they change conformation, does the active site now able to bind with a substrate? The answer is no, cannot. And does they block the substrate bind with the active site because of competition? No, no competition is occur. They just simply change the conformation of the active site so that substrate cannot bind with it. So this kind of inhibitor, we are call them as non-competitive inhibitor. So the effect of the inhibition, can we overcome the effect of the inhibition? Okay, so the answer is yes for the competitive inhibitor and no for the non-competitive inhibitor. Okay, so why we say that it is yes for the competitive inhibitor? So just now we said, what is the characteristic for competitive inhibitor? Competition, right? So when I have a lot of substrate, example, where I have a lot of substrate and I have only one inhibitor, two inhibitor. So who will going to win the competition? I mean, successfully to enter the active site? Definitely is the substrate, because we have more substrate compared to inhibitor. So in order to overcome the effect or reduce the effect of the inhibitor, we can actually increasing the concentration of substrate. However, if I have the non-competitive inhibitor today, non-competitive inhibitor is changing the active site, the conformation of active site. So there is actually we didn't have any way to overcome the effect of the non-competitive inhibitor, and the only method is to let the inhibitor detach from the enzyme. If you cannot actually let the inhibitor to detach from the enzyme, then there is actually no other solution. Okay, but in case if you really want the rate of the reaction to be increased back, then you can try to increase the amount of enzyme by adding the new enzyme. But if let's say you don't have the new enzyme, definitely you cannot overcome the reactions of the non-competitive inhibitor, so which is actually being written down here. Okay, so you can read through by yourself the detailed explanation, but I just simply go through why they are called competitive, because they have similar shape or conformation with substrate. They have a competition to bind with the active site, that's why they are called competitive inhibitor. Does it able to overcome? Yes, by increasing the substrate concentration. So this is how they overcome it. And can you actually identify it through the graph? The answer is also yes. With the presence of the inhibitor, you're going to see that they're actually taking a longer time for the or higher substrate concentration for the rate of reaction to reach the optimal level. So if let's say today you have the non-competitive inhibitor, you're going to notice that the optimal level is decreased. You cannot actually reach the original optimal levels of the optimal rate of the reaction, but the rate of reaction definitely is reducing. Why? Because increasing substrate concentration do not help to overcome the effect of non-competitive inhibitor. Okay, so what is the example? The next thing, what is the examples for the competitive inhibitor? We have the melanoid, melonic acid, which is the inhibitor for the succinic acid. The enzyme involved here is a succinate dehydrogenase. Both of them have the same shape. So melonic acid, the presence of melonic acid can block the succinate acid from binding with succinate dehydrogenase. Okay, where else for non-competitive inhibitor, we have the isoleucine. Okay, isoleucine, which eventually we're going to inhibit the threonine deaminase to run their function. Okay, comparison between these two inhibitor is actually present in this table. So try to actually make it become a complete statement. This is a point form. So later on, I hope that everyone can make it become a complete statement. And then that's it for the reversible inhibition. So they are temporarily bind. These two, they are temporarily bind with the enzyme. At some time, it will detach and the enzyme will remain back to their own function. Okay, irreversible inhibitor, which is different with the compared with the reversible inhibitor. Irreversible is permanent. They are permanently bind with the enzyme. So their effect is permanent. They will damage the enzyme permanently, causing the enzyme to lose their function. So those irreversible inhibitor involve heavy metal like the mercury, silver, which is always involved in the water contamination. And then we have also the cyanide, which is one of the chemical substance that used widely in the industrial. So all this is actually considered to be irreversible inhibitor, especially for cyanide. If a person is exposed to certain amount of cyanide, it will going to cause all the mitochondria to fail to carry out their function to synthesis ATP, and it will immediately kill the person if, let's say, the dose of the cyanide intake is too high. Because this is irreversible inhibitor, we can almost say that if the person accidentally consumed too much of cyanide, there's actually no cure for the person, because the effect is immediate for cyanide. Okay, others, heavy metal ion. Okay, heavy metal ion, extra information for you lah. They do not actually cause an immediate effect like cyanide. Cyanide is very immediate effect. Immediately, the person will feel when the overdose of the cyanide is present. But heavy metal can accumulate in our body system. Even though after they bind with the enzyme, they are permanently damage the enzyme. But our body system seems to have the speed to actually cover back the amount of enzyme damage with new enzyme. However, the remain damaged enzyme is not able to break down by the body cell, and the heavy metal will continue to stay inside our cell. And when the accumulation of the accumulation is actually happen, then level by level it will actually